Hello all, welcome back to our lecture. This time, today we'll be talking about the three-dimensional structure of proteins. This will be the first part of our module 4 of our chapter 4. Let's take it off from there. What are proteins? That's the first question we need to answer. Proteins are polymers of amino acids that are held together by covalent bonds. So polys, you can just say they are polypeptides. When you have a lot of peptides, of course, peptides like we talked about in the last chapter are held together by that peptide bond. So when you have a lot of these guys held together by these peptide bonds, long chains of this amino acid held together by peptide bond, that's what we call what? They are proteins. Of course, remember that proteins run from the N terminal to they go to the alpha amino acid to the C terminal. The C terminal, usually the carboxylic acid terminal. Do not forget that. So many conformations are possible for protein due to the flexibility of these amino acids. Now, that will lead us to what we call the native conformation. The native conformation is the three-dimensional shape of a protein with biological activity. Now, if a protein has its typical shape, it's likely going to have its activity. Because when these shapes are unraveled or broken, when those bonds holding them together are broken, it loses that particular shape. So every protein wants to be in its native. The native structure means it's a healthy protein having all its three-dimensional structure. That is a protein that is going to have a biological functions. So of course, many proteins have large segments of random structure as they have no obvious repeating structure. They have obvious repeating pattern, which is the amino acid pattern, but the structure may not be very obvious because those amino acids can fold on each other and turn in different dimensions, as we're going to see in their 3D. So what are the functions of protein? Before we move on, let's talk about the functions of protein. Now, the proteins have versatile functions. You cannot have enough of what the protein functions are, but we're going to look at a few here, and it's important you need to know these functions. And anytime you are giving me the functions of proteins, it's important you also do what? Give me examples like I did here. So most proteins are enzymes. 98 to 99% of proteins are enzymes. Good examples are the lactate dehydrogenase and the chymotrypsin. They can be regulatory in nature. They regulate both metabolism and gene expressions. Those ones that play a role in regulation are usually the hormones. Example, the insulin and the glucagon. They can also play a role in transport. Transport proteins can be the sodium potassium ATPase that we looked at earlier, the plasma proteins, the hemoglobins, and the lipoproteins are all the lipoproteins like the LDL and VLDL are all HDL. They're all transport proteins. Proteins can also store other amino acids or they can store other nutrients. A good example is the ferritin. Ferritin stores iron. All right. So other protein functions include movement. Movement as achieved by the contractile proteins in actin and myosin of the muscle. Proteins can also be structural in nature. Example is the collagen triple helix. We can find in most connective tissue, and even keratin that we find in the hair. Other proteins can also have protective and exploitative function. Example is the immunoglobulins. The immunoglobulins and the antibodies are good examples of proteins that play an important role in protecting the body against invading organisms. And other proteins a few proteins can have exotic functions. Exotic function, a good example is the antifreeze proteins that are found in the teleost fish help this fish to survive against harsh frozen weather conditions. So these are a brief look at the functions of proteins. So what are the levels of protein structure? Levels of protein structure. There are 4 levels of protein structure. If we are to understand the native or the three-dimensional structure of protein, we have to look at it from the simple structure to the complex. The simple structure of proteins are the ordered amino acid arrangement in a covalent bond. Of course, it runs, like I said, it runs from the N terminal end to the C terminal end. That link of chains of amino acid connected from NCC is what we call the primary structure of course, we're going to see how important it is because it is the foundational structure of protein that determines its three-dimensional structure. The secondary structure of protein is the other three-dimensional arrangement that is based on hydrogen bonding from the polypeptide backbone. It does not involve the arrow group. However, by the time we get to the tertiary structure, we now begin to see that in the tertiary structure is the three-dimensional arrangement based on the side chains. The side chains will be like, remember when we talked about the different amino acids, we classify them based on their side chain. The side chain gives them identity. They can be basic, they can be acidic, they can be polar, neutral, they can be polar charged, they can be non-polar as well. So we're going to see the importance of those side chains on the tertiary structure. And the quaternary structure simply means you have a particular protein that have more than one subunit. More than one subunit, you have more than one polypeptide chain. That's what we mean. So simply here, you can just say, in this kind of, you have more than one polypeptide chain, more than one polypeptide chain. Now, when these polypeptide chain fold on each other and interact, what happens? They also have those, they also form those negative structures and have those bonds that stabilize both the tertiary structures. And in fact, the same types of bonds stabilize both the tertiary and the quaternary structures. All right. So a subunit simply means an individual part of a large molecule. Like I said here, each polypeptide chain is called what? It's called a subunit. So let's now begin to take them one after the other. Amino acid, primary structure. The primary structure of amino acid is just the amino acid sequence as determined by the genetic code. The genetic code is what determines how this amino acid are sewn together. So I look at it as a jewelry chain or a bead, a jewelry bead that has each of the beads sewn together through a long group. That is exactly what the primary structure looks like. Of course, it determines what? The three-dimensional structure of proteins. It is what determines the three, because depending on what the amino acid looks like, if it's a polar amino acid and it's going to interact with another polar one, there's possibility of forming a hydrogen bond. And if you're going to form a hydrogen bond in tertiary structure, that is going to change. That's going to bring about the type of conformation in that tertiary structure. So the determination of this sequence of amino acid is a routine biochemical operations. And it's very, very important because removal of one amino acid for the other will lead to mutation. And any mutation of a single amino acid can change not just the biological function of that amino acid, but some of them can lead to very serious diseases. A good example is that sickle cell anemia is a disease whereby there is a mutation of glutamic acid being replaced by valine. This single replacement of this amino acid causes enough problem that the hemoglobin of a sickle cell patient is not as effective as those, is not as effective in carrying oxygen when you compare that to that of a healthy person. And that could lead to seriously debilitating consequences in the sufferers of sickle cell anemia. So the primary structure of protein is exemplified. What I have here is the bovine pancreatic ribonuclease A. If you look at this now, if you look at this bovine, of course, I told you that you remember this by now, that the polypeptide chain runs from the amino group, amino N-terminal, goes to what? The carboxylic group. Now, it runs here, of course, you see it, let me not cover that, okay, yeah, it runs from N to here, and it has 124 amino acids. And if you look at it, it has 4 cysteine groups, 4 disulfide groups that you see between the cysteines, holding them together. So, if you look at the 4 diphosphoride bridges, so if you count this long arrangement of this amino acid, that is exactly what we mean by a primary structure. So the primary structure is just that long sequence of attachment of amino acid alone. We just talk about the polypeptide bond. So the peptide bond is what stabilizes the primary structure, just the polypeptide bond. We go to the secondary structure. The secondary structure is stabilized by hydrogen bonding between the carbon of one peptide and the forming a hydrogen bond to the amino group of another amino acid. And this hydrogen bond is strong enough to maintain a steady structure that are of two types, the alpha helix and the beta-plated sheet. Now remember, it's important to say this, that the secondary structure does not involve the arrow groups yet. It still happens only in the polypeptide chain, and this can give rise to two types of structures. Now the alpha helix, oh, that helix doesn't look beautiful. The alpha helix looks like a helix, like a spring helix. Now in this helix, the polypeptide chains, let me use a different color, are held together in these chains. Remember again, this is what we mean. This is the primary structure is being held in this chain. These are the amino acid. I'm not going to go all the way, but I'm going to show you. Now look at what happens here is that hydrogen bonding can occur between any of these. Oh, I'm going to use a different color then because that's overlapping. I'm going to perhaps use blue. So hydrogen bonding can occur between these two. Hydrogen bonding can occur between these two. Hydrogen bonding can occur between these. This hydrogen bonding is what maintains the structure, giving it this alpha helix. Is what gives it this alpha helical structure. This is the alpha helix, like a helix. And then the beta-plated sheet, on the other hand, will have amino acids that are running parallel or anti-parallel to each other. So this is what happens here. So you have amino acid here in the chain. It has the primary chain running across. Now what happens here is that there will be hydrogen bonding between these guys, between this guy, between this guy, between this guy, holding them together to form what we call the beta sheet, or you say the beta-plated sheet. The beta-plated sheet. These are the two structures that we see, and we're going to see this more in the next few pages in a better structure. So now this is the alpha. Again, remember that the polypeptide chain will run from the NCC, from the N terminal to the alpha carbon to the carboxylic. Now the NCC, look at the NCC. NCC runs and folds in a helical manner. This is a short strand, and they can now form this hydrogen bond within this polypeptide chain. Again, this is the beta-plated sheet, like I showed you earlier. In the beta-plated sheet, it's just the sheet running on its own, and they can form the structure within themselves. So let's talk about them here. Now the alpha helix is stabilized by hydrogen bonds that are parallel to the helix axis within a polypeptide backbone. Of course, I said this earlier. I also said that carbon to oxygen, which is a carbonyl bond of which peptide is hydrogen bonded to the NH of the fourth amino acid. It runs from first to four, or some books will usually tell you 3.6 residues of amino acid per ton. It's not a perfect calculation. Some texts will say 4, some will say 3.6. So the helical conformation allows for a linear arrangement. It provides maximum bond strength and makes the conformation stable. So the coil usually runs from clockwise or right-handed. And the distance between each of those turns measures about 5.4 Armstrong. So we're going to, this is again a better, I think this is a better picture. I like this one better. Now this is again NCC, NCC, NCC. And if you look at the NCC running parallel with another one, this is carbon. This is the amino acid, the nitrogen to hydrogen of another one, forming the hydrogen bond. So this is a very good structure to depict. And if you look at this now, there is from here to here, it tries to tell you you have 3.6, or you can say 4 amino acid. And I wouldn't bother you with trying to know the exact stoichiometry of the number of amino acids. Just know that this runs in this helix and forms this hydrogen bond with one another. All right, so the beta-plated sheet is hydrogen bonds, again stabilized by hydrogen bonds. So it's hydrogen bonds between peptide chains that can be intra-chain. Now, it can be, the same chain can run on its own, or it can be two chains running parallel to each other. It can be. But however, they're going to form these interactions. That's what matters. The hydrogen bond interaction has to form in between those amino acids to keep them together. Now this results to this a zigzag structure, and of course polypeptide chains lie adjacent to one another. If the chain runs in the same direction, it could be parallel or anti-parallel. Parallel is when it runs. This is anti-parallel, anti. Then when it runs in the same direction, it is parallel. Remember, the arrow groups do not get involved again. They just protrude out from this arrangement, of course. Now, this is a very good way to show it. So this is the parallel sheet where they are running in the same direction. Assuming we believe that this folded on each other here, comes and folds on each other here and runs in that parallel. Remember, the carbon is always black, oxygen is always red, hydrogen is white, nitrogen is blue. So you see these are hydrogen bonds from these two chains holding them in this structure. This is also in anti-parallel arrangement. And again, like I said, this one shows it clearly. The arrow groups, this is the arrow groups, point away and they don't get involved in the two types of secondary structure. It is just the hydrogen bond as shown by red dotted lines that is our interest at this point. So the helix of a spider web is a mixture of both there. If you look at it, this is the sheet and the helix. You see the helix is not really very clear coming together. So most times it's a mixture and each of them have something they need to offer. All right. Now, the beta sheets tend to be very tough and strong, whereas the alpha helix offers flexibility. The helix offers flexibility, and the combination of these two will, make the spider web very dangerous. That is why the spider web can be used to enslave other smaller insects. Now, the collagen triple helix is another interesting type of protein. Very strong structural component in most connective tissues. It's organized in a water-soluble fibers of great strength. The fibers of three polypeptide chains are wrapped around each other in a Refolding triple helix. It's like a helix. Now that helix is actually called the triple collagen. It has a molecular weight of 300,000. Now each of these chains is a repeated sequence of 3 amino acids that runs from any amino acid. X can be any amino acid, but it must be proline to glycine or any other amino acid. If it's not proline, it can be hydroxyproline. Hydroxyproline is produced from proline in what we call post-translational modification. You just adding an OH group to a proline makes it a hydroxyproline. So it's usually formed after the proline is made in protein synthesis. So the first position of X, like I said, can be occupied by any amino acid, but every third position must be occupied by glycine in the triple helix, making it a very strong. So look at the triple helix. So all the 3 collagen helix chains, all the 3 individual collagen chains are helixes that differ from, they look different from alpha helix, the way they look like. They're held together by hydrogen bond involving the hydroxyproline and the hydroxylysine. Again, hydroxylysine is produced from lysine when it is hydroxylated by a hydroxylating enzyme. Now, with age, collagen helices become even stronger. They cross-link more by covalent bonds formed by the reaction between lysine and histidine residues. So now, this great strength is what makes collagen to be a very tough structural component in our collective tissue. So now, we can also, coming from the secondary structure of protein, we can see 2 types of protein in this case. The fibrous protein or the globular protein, depending on the shape. Now, the fibrous protein contains polypeptide chains that are organized approximately parallel along a single axis. That's exactly what they are. They are in a single axis. They consist of long fibers of flat sheet and they are mechanically strong. When you see fibers are strong. Of course, they are insoluble in water and have the ability to dilute salt solutions. They play important structural roles in nature. Good example of where we can find this is we can find keratin in hair and wool, and we can also find it in collagen. Collagen in connective tissue, including the cartilage. You can find it in the bones, in the teeth, in the skin, and the blood vessels. These are places we can find strong fibers, because fibers are really very strong and form part of the structural component of the body. On the other hand, the globular proteins, from the name, they form globules. What they simply do is that they fold back on themselves and look like a spherical or circular shape. So if you have a protein that runs and folds back on itself, and the importance of this is that they fold back on themselves so that they can form compact structures that can fit into cells. So most of the arrangement is based on the micelle. Remember in chapter 1 we talked about the micelle. In the micelle, the polar sides are outside, interacting with the aqueous environment. The polar chains are outside, interacting with the aqueous environment by hydrogen bonding and other ion dipole interaction. The nonpolar cell chains are buried inside, away from the aqueous environment. Of course, they have substantial portions of both the alpha helix and the beta sheet, and they tend to be soluble in water. And this usually represents the structure of most enzymes and hormones, because most enzymes and hormones are found in the cell, and they need to fit into the cell. That's why their nature gives them that globular shape to fit into the cells. And because also they work in aqueous environment, they want to interact well in forming this micelle. So this is exactly what you see for the enzymes. Most enzymes and hormones are globular proteins. That will lead us to seeing what they look like. So this is a filament, a twist of a filament you can see in a fiber. This is a fiber you can see for a connective tissue. It looks like a long spherical strong tube. Now this is a myoglobulin, which is a globular protein. So globular proteins is that they fold around on each other. This is another picture showing you how they fold on each other. And each of them, like I said, particularly for the globular proteins, they're going to have a mixture of what? They're going to have a mixture of both the alpha helix and the beta-pleated sheet. Tertiary structure of proteins. The tertiary structure of proteins represents the three-dimensional arrangement of atoms. All the atoms in a protein molecule, and includes the conformation of the side chains. Remember, the arrow groups now come into play. Remember, the primary structure held together by what? Peptide bonds. The secondary structure stabilized by hydrogen bonding. The tertiary structure includes every other bond, including all the bonds in the side chain, depending on the identity of the amino acid. And that also includes what we call the prosthetic groups. The prosthetic group is a non-protein portion of the amino acid that helps the protein in its structure and its function. So it is the non-protein part. Because for you to have a native protein, it includes the prosthetic group. It is a non-protein part of the protein. In fibrous protein, the backbones, of course, do not fold in each other. So the fibrous proteins are better understood by their secondary structure. They don't have an extensive tertiary structure. However, in globular proteins, remember what we said, in globular proteins, now they tend to have substantial portions of both the alpha helix and the beta-pleated sheet. And the implication is that they fold on each other. And as they fold on each other, the side chains makes new interactions. Those interactions between the side chains play an important role in those foldings. Those interactions between the side chains play an important role in the folding of the globular proteins. And in fact, the globular proteins are better understood by their tertiary structures. And we're going to see what types of bonds that are involved. Now, the bonds that stabilize or that hold together these tertiary bonds are the following. Number one, it could be hydrogen bonding, can play a role between the polar side chains. Polar side chains are like the acidic amino acid and basic amino acid, and then which interacting with polar neutral ones like threonine, serine, or tyrosine. So those are polar side chains. It can be hydrophobic. Hydrophobic side chains will involve leucine, valine, and alanine. It can be electrostatic. Electrostatic will be between two charged opposites. Like if you have lysine that has ammonium ion charged and acidic amino acid like glutamine that has the carboxylic group, they can interact and form electrostatic interaction. Metal ion can also form a bridge between two proteins, and that's what we call the metal ion complexation. And we can also have disulfide bonds between cysteine side chains. Now, this is also a type of covalent bond. It restricts folding because it's a covalent bond. Restrict folding pattern available to polypeptide chains. It is, however, absent in myoglobulin and hemoglobin, but present in chymotrypsin and trypsin. So we're going to see this graphically. What does it look like? Now, this is a typical structure that has both the primary, the secondary, and the tertiary. And remember, the primary structure is the long chain of the amino acid. Now, this is the alpha helical portion. This is the beta sheet. Of course, these two are stabilized by hydrogen bonds. Remember that? We talked about it. The hydrogen bonds is what plays role, HB. Now, let's look at other structure. You can have a metal ion in between two proteins helping to stabilize the proteins. This type of bond is what we call the metal-ion coordination or complexation. You can also have hydrophobic bonds can form between non-polar side chains like isoleucine, valine, or leucine. You can have those interactions. You can have disulfide bridge can be formed between two cysteine groups. Two cysteine groups, remember this is cysteine. If you have two cysteine group, arrow group here, they can form this bond. This is what we have, disulfide bridge or disulfide bond. You can also have the polar side chains. Remember, carboxylic group could be from aspartate or glutamate, having the carbon and carbon interacting with that of another one. Here could be a threonine, a serine, or tryptophan that has the OH group. So this forms a hydrogen bond. And again, you can also have electrostatic interaction. Electrostatic is an interaction between two opposite charged entities. Here we have the carboxylic group that could be from any of the acidic amino acid and the ammonium group that comes from any of the basic amino acid. The interaction they have is what we call the electron. So these are the bonds that stabilize the tertiary and actually the quaternary structure of proteins. Now, determination of the structure of protein is done through many ways. Particularly, the X-ray crystallography is used to determine the structure. Here, it uses a perfect crystal in which individual protein molecules have the same three-dimensional structure and orientation. So when this pure crystal is exposed to the beams of X-ray, a diffraction pattern happens. This diffraction pattern can be constructive or destructive. The constructive ones is the one that is analyzed and fed to Fourier series where it is analyzed and we can get the structure of the protein. You can also do that with the nuclear magnetic resonance spectroscopy. In 2D NMR, large collection of data points can be subjected to analysis. Again, you use the Fourier to analyze this and then come up with what the structure of the protein will look like. So this is a typical example of the tertiary structure of lactalbumin. Lactalbumin here, of course, like I told you, every amino acid will run from the N group, this is the 2, the carboxylic group. And you see when it runs all through from this N group until it finishes on this one. And if you find that this is a mixture of all of them, it has, let's see what it has here. It has sections of the alpha helix about 4 places, and it also has only about 1 section here of the beta-pleated sheet. Remember, this is the beta-pleated sheet. Let me just say the beta sheet. The beta sheet, and this is what? This is the alpha helix. And then remember again, as it's folding on itself, it makes those the other interactions that are stabilized. Those other interactions that stabilize the tertiary structure. So this is the typical way that you see your protein existing, having a mixture of all these structures to put them together to form the three-dimensional state, or what we call the native state. And it is this native state that has both the true functions of these proteins. Let us now look at denaturation and refolding. Denaturation simply means the unraveling or the breaking down of this three-dimensional structure of any macromolecule. This time we're talking about protein. And what happens here is that those non-covalent bonds in the tertiary structure, remember, those non-covalent bonds in tertiary structure are broken down. They are broken down. And then the protein loses its three-dimensional structure, and hence it's what? It's not only the three-dimensional, but it loses its function. And what are those things that can do this? They include the heat can do this. Heat can do this. Remember, enzymes or proteins have a tight pH range in which they can survive. Either too low or too high is going to be disastrous to them. Now, changes in pH, they also have a tight pH range, about 7.4, anything above or below, or not generally, it depending on what protein they are, they have a particular pH range in which they act optimally. So going away or above that is going to be a serious problem. It can be detergent like sodium docile sulfate, SDS, can disrupt the hydrophobic interaction. Urea and guanidine hydrochloride will also disrupt the hydrogen bonding. And then Beca-mercaptol 2 is a strong detergent that can also disrupt all this. So all these exposures will render the protein useless when it loses its tertiary structure. So this is the native state when it can function the way it wants to be. Look at it, it has both the alpha, this is the alpha helix here. When you break those bonds that hold them together, it forms just a long chain. And in this nature, it has lost its function. It has lost its function. Because every protein, to be good, for it to be stable, it has to have its function and structure at the same time. So this is also denaturation and refolding of the ribonuclease. The ribonuclease here, you see, Beca-mercaptol is used to reduce the disulfide linkages. That it has the disulfide here, 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 and here. This is used to break the disulfide. Urea is added to facilitate the unfolding and then increasing accessibility of the disulfide bond to the reducing agent. Now, if you do all these things, what happens now is that after addition of both the Beca-mercaptol, you look at what happens here. It breaks this bond, the disulfide bond, and it loses its native structure and becomes this. When this unfavorable environment is removed again, the protein itself tends to reform naturally, provided you remove those things and you don't break the primary structures and break the amino acid into single. Provided the amino acid are still intact in the primary structure, when you remove that unfavorable condition, when you remove it, it can refold itself and form back to a normal structure. So this is a typical example of what you do every day when you eat your egg. So the egg contains this, the ovoalbumin. So when you begin to fry your egg in high heat, what you do is that you denature this ovoalbumin in high heat and render it useless, and this is what you eat. So the figure above shows protein of egg white that are denatured during cooking. More than half of the protein in egg white represents the ovoalbumin. And with this being said, we're going to be coming to the end of this lecture. Once again, stay safe and thank you for listening and have a wonderful day ahead. And bye at this point.